When we think of scientific discovery, we picture sterile environments, controlled variables, airtight protocols, and gleaming stainless steel, where every outcome is precisely managed. In 1928, Alexander Fleming operated differently. He was a highly competent bacteriologist, but his habit of leaving used Petri dishes stacked in the sink routinely irritated his more methodical colleagues at St. Mary's Hospital. His second-floor workspace was not a hermetically sealed environment. It was an untidy collection of mismatched glassware and exposed equipment, where outside spores drifted freely through the building. In this pre-antibiotic era, a simple scratch from a rose thorn often led to lethal blood poisoning. Infection was the leading cause of death from injury, surgery, and childbirth. The cure for these infections, a drug credited with saving over 200 million lives, depended entirely on a specific sequence of negligent errors. This is a petri dish showing the Staphylococcus aureus colonies Fleming was studying. Standard practice was to autoclave these dishes immediately after use. Fleming instead stacked them in his sink and left them. In the summer of 1928, he abruptly abandoned his uncleaned laboratory to go on a 2-week vacation. The lab directly below Fleming's happened to be studying mold samples. Because the facility was not airtight, fungal spores routinely drifted upward through the building and into the second-story lab. A single spore of Penicillium notatum landed on one of the unwashed dishes in the sink. It was left undisturbed to grow for 14 days. A perfectly maintained, protocol-compliant laboratory would have sterilized the dish or filtered the air. In a clean environment, this discovery would have been impossible. Returning in September, Fleming began sorting through the contaminated plates he was about to discard. He noticed a blue-green mold had grown on one dish. Remarkably, the Staphylococcus colonies nearest the mold were gone, completely dissolved. The bacteria were being lysed. Their cell walls were breaking down and dissolving because of a chemical the mold was producing. Fleming's reaction was famously understated. That's funny. He decided to keep the contaminated dish and culture the mold, which he eventually named Penicillin. The accident provided the raw data, but the discovery required an observant mind prepared to investigate an anomaly rather than discard a failure. Fleming could not stabilize his mold juice. In its raw form, penicillin was too impure to be used as medicine and degraded too quickly to be manufactured. After publishing his findings in 1929, Fleming largely moved on to other research. For a decade, the discovery remained a dormant scientific curiosity. In 1938, Australian pathologist Howard Florey began a new survey of natural antibacterial substances at Oxford. His colleague, biochemist Ernst Chain, searched the old literature and unearthed Fleming's 1929 paper. Chain provided the chemical expertise necessary to solve the purification problem that had stopped Fleming. The published paper almost remained forgotten in the academic literature. It preserved Fleming's observation for 10 years until a new team with the right motivation and skills could develop it. As World War II began, finding a cure for battlefield sepsis became an urgent military priority. The war provided the incentive to move the drug from the lab to the front lines. The 1941 trial of Albert Alexander proved that the drug worked, but the lab's small yield could not save him. He improved dramatically for days, then relapsed and died when the supply of penicillin ran out. To reach industrial scale, Florey traveled to the United States to enlist pharmaceutical companies. Pfizer, Merck, and Squibb were recruited into a crash mass production program. This ampule represents a logistical triumph. By the D-Day invasion in 1944, the Allies had enough penicillin to treat every major infection among the troops. In 1945, Fleming, Florey, and Chain shared the Nobel Prize, acknowledging that the cure required all three men's contributions to succeed. The forgotten Petri dish provided the initial accident. Translating it into 200 million saved lives required the observant mind to see it and the industrial infrastructure to scale it to the world.